Everybody, it's Mr. Alex, and I'm here to talk about DNA replication. So this is an exciting topic if you are interested in genetics and the way that our body makes us look and feel and act the way that we do. This is true for all organisms. The central dogma, we call it, really begins with DNA replication. So looking at how our DNA is replicated helps us understand what these little enzymes are that are doing the replication process, and then lets us understand how our DNA gets transcribed or copied into RNA. Now that RNA leaves the nucleus of a cell and goes to our protein builders, the ribosomes, to make the proteins that will structurally become part of us, that will be our enzymes, that will give us the phenotype that we have. So we start this central dogma with DNA replication. And here it kind of is in a nutshell. So what we're going to find out in a minute is there's really two active enzymes in this process, helicase and DNA polymerase. We have a double-stranded piece of DNA. We are going to unwind it with helicase. We are going to add a new single strand to both sides using polymerase. So DNA polymerase, it makes polymers, so we call it DNA polymerase. And then we're going to end up with two new double strands of DNA. And you'll see that when we look at each double strand, they each have one old strand from the original template, and then they have one newly formed strand. So when we look at this topic, we sum it up with we're doing DNA replication to produce exact copies. That type of replication I just described is unique to a huge group of organisms on our planet, and it's called semi-conservative. We'll talk about what that means again. We'll look a little bit closer at those two enzymes I described that do DNA replication, and we'll look at a really cool application. So when we want to compare two people's DNA or look at the DNA among species, we can't just take a small DNA sample. We need to take that sample and then copy it and copy it and copy it and do what we call amplification through PCR, a polymerase chain reaction, and then we can analyze it through gel electrophoresis, which is actually a pretty old method. We have more advanced methods now. And that lets us compare forensic evidence at a crime scene, look and do maternity and paternity testing, or ancestry analysis, or looking at the data related to, or the base sequence, DNA-based sequences of species that might go extinct or that are extinct. So that's where we end the SL content. We move on a little bit more to how replication is happening on each of those strands. We have nicknames for them, the leading strand and the lagging strand. A little bit more about what's going on with those enzymes, and kind of a deeper dive, especially on DNA polymerase. There's actually not just one, there's three. HL people are expected to know what 1 and 3 are doing, and it actually helps us understand the process. So if you're HL, you get to go a little bit deeper here. So let's get started. So DNA replication, we're making two exact copies of one piece of double-stranded DNA. When we look at it, we have that original template that actually becomes part of each of the two new double-stranded DNA molecules that we form. So that single strand from the original template becomes part of this double strand. This single strand of that original template becomes part of this double strand, and we also get a newly synthesized strand as part of that. If you're not familiar with the structure of DNA, we have already covered this. So at this point, if you're not, pause, go back to nucleic acids, review the structure of a nucleotide so that you understand what's being added when these enzymes are working. It'll help you understand what can go wrong, how the molecule itself is bound. I'll give you a real quick synopsis now, but you should be familiar from that topic. A nucleotide is made up of a phosphate group, a sugar, and a nitrogenous base. In DNA, we have 4 nitrogenous bases: adenine, thymine, cytosine, and guanine. C pairs with G, A pairs with T. And this is the representation we use in the IB of this molecule. You can look deeper and see where the actual bonds and atoms are, and I recommend that you do that. That will help you understand why we call this the 5' end and this the 3' end. I'll talk a little bit about it later on, especially for HL students. But when we look at this nucleotide as part of a big DNA polymer, right? So this is DNA. This is one DNA nucleotide. It is double-stranded. It ends up having a helical shape once everything is bonded. It is anti-parallel, meaning one side goes up and one side goes down. So it's a very directional molecule. This is nice. The two strands don't have to be different, right? They're actually the same order, the same chemistry. They're just facing in different directions. So we don't have to make DNA differently, but when it comes to replication in this topic, we'll learn that you do have to copy it a little bit differently when one strand is facing the other way. The nucleotides I described a moment ago, and the complementary base pairing. DNA nucleotides, this sugar is deoxyribose. In RNA, this sugar will be ribose, so it has an H here in deoxyribose, so it'll have an OH in ribose. That little difference matters. RNA is made in a complementary way, but exists as a single strand, and DNA exists as a double strand in humans. You can have double-stranded DNA, single-stranded DNA, double-stranded RNA, single-stranded RNA. In other organisms, in us, we have single-stranded RNA and double-stranded DNA, which we're used to seeing. We see that double helix. Like if I look at this image of replication, it's like, wow, there's a lot going on here. But follow with me. If we look at the original strand, we see double-stranded DNA. We see our buddy helicase, which is going to be responsible for splitting that strand, unwinding it essentially, breaking the hydrogen bonds in the center, not breaking any real bonds, just pulling apart the hydrogen bonds, which are not actual bonds. They're just forces of attraction. And then polymerase, these DNA polymerases shown here with this kind of like taupe, teal-shaped C, go ahead and copy or add that newly synthesized strand to each of the original single strands. And we end up with two perfect copies of that original. We need to do replication to grow and replace our tissue. We'll also do it prior to meiosis and reproduction, but generally right now we're talking about replication with regard to mitosis. But this is a vital process for life. So that type of replication that I've been describing is called semi-conservative. And the reason it's called semi-conservative is because we conserve part of that original DNA strand, right? So semi. We do make a newly synthesized strand. That's what our DNA polymerase is going to do. It's going to add nucleotides to make that new strand. But that original strand remains in part. So we call it semi-conservative. So if we look at the original strand in red, notice that it is conserved in each of these, but not fully conserved. The newly synthesized strand is added. If it were fully conserved, we do see this in some organisms, it would look like this. There's also dispersive replication, but we are talking about semi-conservative replication. We had a great HL IB Bio question where they showed a diagram and they said which one shows how DNA is replicated. And they wanted students to choose the semi-conservative replication. The way that they showed it too was very interesting, and we'll look at that in class. So if we want to think about how DNA bases get added, we can think about the structure of those nucleotides. The red pentagon being the sugar, the bases here, and then the phosphate group there at the end. What's actually happening is DNA polymerase is in charge of letting these bases line up, and then its really important job is making that covalent phosphodiester bond. This hydrogen bond will happen on its own based on attraction. It kind of helps the whole process. G will line up with C, C will line up with G, but that DNA polymerase's job is really to make that bond. And the complementary nature of DNA and of these bases make it easy to assemble those. So if we look at a really simple representation of our enzyme that assembles the nucleotides, our DNA polymerase, and our enzyme that unwinds DNA helicase, this is what it might look like. And we run into this situation that we'll examine in HL a little bit deeper, where because DNA is anti-parallel, and the strands face opposite directions, DNA polymerase only copies, or only replicates, I should say, right, in one direction. So after helicase splits, or as helicase is unwinding DNA, one DNA polymerase can just be working. It can be adding nucleotides happily to one strand, making a new double strand. But because the other one is facing the other way, that DNA polymerase has to work sort of away, then stop, and then come back, and then work away, and then stop, and then come back. And so we'll look closer at how that process works in HL. But I have a couple really good videos to kind of get you started. I recommend you pause and kind of check these out. This one is really beautiful. We'll look at this in a minute. This really lets us look at how
using computer animation based on molecular research, we are now able to see how DNA is actually copied in living cells.
Two DNA polymerases on either side.
You are looking at an assembly line of amazing miniature biochemical machines that are pulling apart the DNA double helix and cranking out a copy of each strand.
On this other side, where because this one is copying
the DNA to be copied enters the production line from bottom left.
Stop and start again.
The whirling blue molecular machine is called helicase.
And you'll see as this
it
DNA
spins the DNA as fast as a jet engine as it unwinds the double helix into two strands.
Kind of where it came from, or rather
one strand is copied continuously
so it needs to keep,
and can be seen spooling off to the right.
Along that strand and beginning again. Yeah,
things are not so
right away from, and then it's stopping and coming back. So we'll look at that video again in the HL section, but here are these two enzymes that we need to know a bit about. This is a better representation. These are obviously two PDB molecules of the month, so we can see that really nice structure rendered from PDB models, the protein data bank. But we tend to show them really simply sometimes. This is an okay representation of helicase, but I really like this beautiful one. You can actually, if you look in the center, you can see the place where DNA's double-stranded alpha helix kind of fits so that it can split that molecule and bring it apart. When I studied helicase in school, we just drew a triangle, which is not at all what it looks like. Sometimes you'll see it like this. So this is like an ultra-realistic representation. But all these get the point across. It unwinds DNA, it breaks those hydrogen bonds, and enables both strands to be worked on by DNA polymerase. DNA polymerase, I took this image from actually a prokaryote polymerase. Any organism that can replicate DNA has DNA polymerase. Would look more like this, depending on the organism. It's a really diverse enzyme. The top part is really in charge of synthesizing the DNA by adding those nucleotides. But then it can also, we'll see, proofread for errors. And when we separate DNA polymerase 3 from DNA polymerase 1 later, we'll see that they actually kind of have different roles. DNA polymerase 3 is really in charge of replicating DNA. It's the workhorse. Whereas DNA polymerase 1 needs to go back and do something on that strand that we pause on. But that is an HL portion of this, so we'll skip it for now. When we look at DNA polymerase, we also see different representations. You'll see this kind of C-shaped one a lot, which is great. Not all DNA polymerases have this bottom piece, so that's good. You'll see it like this, but it gets the point across. We're adding nucleotides to synthesize a new single strand on that original template strand to make our new strand. And again, you can kind of see these working. The way in this simulation they show
computer animation based on molecular research,
so we can see that
we are now able to see how DNA is actually copied in living cells.
Apparently in real life. It's splitting.
You are looking at an assembly line of amazing miniature biochemical machines that are pulling apart the DNA double helix and cranking out a copy of each strand.
Away from where it's split.
The DNA to be copied enters the production line from bottom left.
And that requires
the whirling blue molecular machine is called.
So when we look at DNA polymerases among organisms, they're similar, but they're not identical. So this simulates what a couple different ones from different organisms would look like. I recommend you go check these out at PDB with some DNA fitting into them. This would be or rather coming out of them after the newly synthesized strand is added. The template strand would be green and the newly synthesized one would be purple. So they do have different structures, and you don't really need to memorize the structure, you need to memorize the function, right? I'm just trying to give you some really relevant and realistic imagery to help you remember that. We'll come back and in HL we'll look at the difference between DNA polymerase 1 and polymerase 2. And just like we saw in that video, you'll see that DNA polymerase 3. 2 is related to repair. All my students always ask, where's DNA polymerase 2? It's related to repair. You need to know in HL the difference between 3 and 1. 3 is the workhorse. It's part of this whole big complex with 2 polymerases, and it's really replicating. It's really in charge of replicating, doing the majority of that
Using
process.
computer animation based on molecular research,
and one
we are now able to
this image. One goes back to the lagging strand and removes what we call primers and adds DNA nucleotides. But that's HL. SL people, you just need to know what DNA polymerase does. And again, it adds new DNA nucleotides. It is capable of removing primers and proofreading. Proofreading means to correct mismatched bases, and we'll come back to that in HL. We will also come back to what is going on that leading strand and that lagging strand. So our great application that we look at when it comes to DNA replication is how we do this in the lab, because we really do need to replicate DNA on a large scale in the lab through the polymerase chain reaction. It is a technique used to amplify small fragments of DNA, so we actually have enough to analyze. We use it in forensics, matching children to parents, so crime scenes, maternity, paternity testing. We use it to analyze or sequence species we think might go extinct, or if we're able to recover, some of a woolly mammoth, maybe we can sequence its data, DNA from a small sample. And the way that it works is pretty ingenious. And for HL people, it will really highlight what these primers are in a minute. So the process is we heat up DNA, we tell the DNA polymerase that's in the machine where to start by adding a tag that says start here called a primer. We then let that DNA polymerase go to work and we get ourselves a copy. We keep cycling this process. In fact, most of these modern machines that are sold are called thermal cyclers, and we end up with a lot of DNA. So for instance, if we look at this short little video,
polymerase chain reaction,
and I'll speed it up. What we'll see is DNA gets heated up and splits apart. Cooled down, and we add the primers, which are tags which tell the DNA polymerase where to begin. The DNA polymerase then comes in at a higher temperature and replicates. And we're good. We have 2 new strands, and we just do it again. So we go from having one original strand to exponential growth of that strand, and we can end up with billions, yes, billions of copies. Because think about how small DNA is. We really need that. So go ahead and review PCR, check that out. My students that are good with enzymes are like, wait a minute, we covered enzymes. That is too hot for a human enzyme to work at. Ours work at 37. This one's working at 73. We need an enzyme in there that can tolerate high temperature, because we want to split the hydrogen bonds at really high temperature. We want to do all of this at really high temperature. So scientists thought of this ingenious solution. They went and harvested an enzyme that naturally lives and exists at high temperatures. Remember I was saying that DNA polymerase is present in a lot of organisms. So Taq polymerase is present in thermally active prokaryotes in like hot springs that live at really high temperatures. So this is actually the one that we use in PCR. We use Taq polymerase to make these copies. There was a IB Bio question that said, tell me about Taq polymerase, and the correct answer was it is a heat-tolerant, heat-stable DNA polymerase that is used for PCR. And here's just kind of a reminder. We start with that one target sequence. We add a primer. We end up with 2 copies. But then there's exponential growth till we get millions or billions of copies. So what's actually in that little vial that goes into his thermal cycler? It's our target DNA. It's our primer tags. It's the nucleotides we need to build. It's the polymerase, the Taq polymerase. We need to assemble those nucleotides and do the building, make the phosphodiester bonds, a buffer to create good situation for building and the appropriate nutrients for that as well. Primers are great. We can use them to select small segments of DNA. We also have restriction enzymes that are used to cut and sort of take different fragments so that we can use them. And we have very specific segments that we'll cut to get our samples. Let's say we want to compare 2 individuals. So once we have all those copies, we can do something called gel electrophoresis. DNA, because it has that phosphate group in the nucleotide, is a polar molecule. So it naturally has a, let's find my gel electrophoresis slides. It naturally has a negative charge. And so what we'll do is we'll take our samples that we made through our thermal cycler, and we'll put them in these little wells, and then we'll run an electric current through it. The DNA will be pushed away from the negative electrode, and it will be drawn to the positive electrode. Now, the DNA will end up being separated in this gel by its size. So essentially, the gel is resistant so that big pieces get caught up early in the gel and small pieces go farther. So what you end up is farther down the gel, you end up with smaller pieces of DNA, maybe 1,000 base pairs long or 2,500 base pairs long. And high up in the gel, you end up with maybe 20,000 base pairs long, so bigger pieces. So they're drawn to that positive electrode, and the smallest fragments go the farthest. So if you see a diagram of this asking about fragments, the biggest ones are up here, the smallest ones are down here. DNA is negatively charged, it's drawn to that positive electrode. And you'll end up with something like this to compare. A lot of these gel electrophoresis samples must be compared under fluorescent light, and fluorescent dye is used. They tend to have a key on the side that shows the ideal sizes of different things, and then the samples inside. Maybe they do a couple trials to make sure the data is right. So we used to do this a lot, but since we've been doing DNA profiling and things like that, we've developed better ways. So in class, we looked at the different applications of DNA profiling that I've sort of alluded to, like forensic science, testing a child's parents, viral bacterial testing to see what you're infected with, ancestry, disease, illegal wildlife trade, species ID, all that stuff. And we're able to compare, in forensics, parents based on a parent's DNA, based on what we were able to get, amplify through PCR, and then generate through our gel electrophoresis. So we have these activities where you can figure out, we did this in class, like who the suspect is or who the parent is, and you'll notice that they always have strands or samples of DNA that align with the child. Or for instance, the specimen found aligns with this suspect. You can see all the bands, the DNA align perfectly. So I recommend you watch one of those videos on PCR. We used to use this electrophoresis, but modern PCR machines no longer need the gel to do it. So the overall PCR process is we get the DNA, we do the PCR, we let the thermal cycler run. We have those 3 phases for separating the hydrogen bonds, adding the primers, and then actually replicating the DNA. We do that over and over again. And then we can analyze it through gel. We can compare the bands. But these days, we have machines that will do this for us. And they're very specialized. So for instance, if you want to use DNA profiling, we have one that's really good for that. Capillary electrophoresis you'd find in like a forensic lab. You're looking for the presence of certain very specific pieces of DNA. Maybe you're doing COVID testing, it'd be this guy. Maybe you want a precise count of the amount of base pairs a species has, you'd use this guy. You're comparing short sequences, you use this guy. And you do not need to know the differences. But I used to teach this, and students would say, wait, like, do we still use the gel? Like, what's going on? And we don't really use it very much anymore, except to learn about this. And I'll come back to that in a moment. Like modern COVID testing, you're not looking at gel. You are looking at bands on a digital readout on a computer with an ideal positive, seen here by this red curve, and an ideal negative, seen here by this black curve. So the image above shows pretty much all positive patients. Yeah, all positive patients. I don't see any blues down here. And down here, many negative patients with you can see some positives aligning kind of near that red band. So that's what we might see. This is what we might see comparing on modern electrophoresis through a computer. When we want to look at genetic markers, we have 24 set genetic markers that we want to look for with individuals, and you'll end up with a readout like this. So we don't use gel anymore for diagnostics, forensics, and sequencing, but we still use it for lab work, and it is a great way to explain how a PCR works and why it works that way and what we're doing and what we get out of it. And that is it for SL. So We'll go deeper into how this happens in a minute, but here was our DNA replication. This little guy symbolizes helicase, unwinding it, DNA polymerase. We're going to see that DNA polymerase is directional in a second. We're going to really highlight that, and on to the HL content. So again, with HL, we're going to examine the directionality of DNA polymerase, what's happening on the leading and lagging strand. We're going to look a little bit closer at all these enzymes, and we're going to talk about proofreading. So let's go ahead and do that. Yep, DNA polymerase works in one direction. So when you have an anti-parallel molecule, this is kind of complicated, right? Like the 3' and the 5' ends of DNA, what that means is when you count the carbons on a ring, something we commonly do in chemistry, you use number notation with these little primes. So what these numbers really mean are the number carbon. So this carbon on a deoxyribose, this little guy right here, kind of the bottom left one, the one on the quote-unquote bottom, is the 3' carbon. That links to the next phosphate, the next nucleotide down. The 5' carbon, there's actually a carbon right in here. Like if we go all the way back up to the beginning when I showed you that nucleotide, this carbon is the 5' carbon. We can think of it as the phosphate group carbon. So if we think about the orientation of a DNA molecule, the 3' is on the bottom, 5' is on the top, but top and bottom are so relative that those words don't even necessarily have meaning in biology, and that's why we use 3', that bottom carbon, 5', that phosphate group carbon. If we flip the molecule, it goes the other way. Now, DNA polymerase adds nucleotides at the 3' end. So when we looked at that diagram before, it is adding right here at the bottom of the DNA strand, what I called the bottom a minute ago, at the 3' side, which means that because the other strand is in the opposite direction, it needs to go the other way. It can only add here as it builds. So it's working on a template strand that goes from 3 to 5 because it's adding from 5 to 3. It's adding on, it's sticking little nucleotides right on here at the 3' end. So that directionality means that replication is going to be really easy on the leading strand. It's going to be continuous, but it's going to be interrupted and discontinuous on the lagging strand. When we look at DNA polymerase, it's also going to highlight the need for 2 different DNA polymerases, in DNA polymerase 1 and DNA polymerase 2. So I made this kind of little GIF here, or my GIF is on the next one, but this just kind of shows us leading and lagging strand to show that the DNA polymerase down here on the leading strand can add continuously and add normally. Whereas on the lagging strand, because of constant interruptions due to it having to replicate sort of away from where helicase is unwinding the strands, it needs to stop and restart. And so this is going to bring some other enzymes into play that are really important. One is DNA polymerase 1. As we add, and I'll go to the one where I made a little GIF, as we start and restart, remember, DNA polymerases, they don't work automatically, and they need a starter tag, like a flag, like, hey, start here. We call that a primer, and it's added by an enzyme called primase. And we can actually see primase here. So primase, every time we restart this strand, needs to fly in and add a little primer tag. It does it right at the beginning of this GIF, like right here, boop, dropped it off. And if we kind of follow this primer tag, we see that's right where DNA polymerase starts building. Those primers are actually made of RNA, which creates a problem. They need to be removed and replaced with DNA nucleotides. So when we look at replication, what is really happening here is this is DNA polymerase 3, the workhorse of replication, this guy. DNA polymerase 1 will need to come in and be the cleanup crew afterwards. It'll need to remove those RNA primers that DNA primase left there and replace them with DNA nucleotides because we keep stopping and restarting the building of that strand. If we kind of really this video is amazing. You'll see primase come in and drop off a primer that'll be used on that lagging strand. You'll see it kind of swoop in here, primer, doot, drops it off. You'll see on the leading strand DNA polymerase, this core working continuously, and this is the core that is on the lagging strand that has to get interrupted and interrupted. So primase keeps on coming in and dropping off those primers so that it knows where to start, but those primers ultimately need to be removed at the ends, and that's what we're not seeing in this video. We're not seeing them get removed. So if we look at all the enzymes that we need to know about in DNA replication, we will see, of course, DNA polymerase, but you'll need to know the difference between 3 and 1. So 3 is the workhorse, it's replicating everything. 1 comes in to remove the primers, the RNA primers, and add nucleotides. Primase was the thing that dropped those primers off to begin with to tell the DNA polymerases where to start. It dropped one off on the leading strand, but it only needs one, just one. Whereas on the lagging strand, it needs like thousands or millions, depending on how big that strand is. Kind of sounds wasteful, right? Helicase is the enzyme that unwinds it. And there's also an enzyme that you should know about. When DNA breakages happen, which can happen accidentally, it can happen when you're sitting in the sun. If you ever smoke a cigarette or something like that and you're exposed to a mutagen, DNA ligase will come in immediately and put the strand back together because we don't want DNA strands kind of improperly assembled anywhere. So this quite is the PDB all-star cast. And those are kind of the guys you should be familiar with. As it's assembling, DNA polymerase 3 can also do something that's pretty amazing. It also does something called proofreading. So let's say in this little GIF that I made, we have the wrong base paired up. Maybe this should be AT, right? Because G pairs with C. That DNA strand will flip down to another part of DNA polymerase and be cut and corrected. So again, we have the wrong part, G. We need to cut that G off. Let's cut that little guy off. Boop. Flip it back up, and we'll add the proper A. It does this as it goes, and you guys saw how quickly this happens. So it's pretty amazing. There are other proofreading mechanisms that happen later, but DNA polymerase 3 is really in charge of proofreading. If it detects an error, the little part on the bottom of the enzyme, we call it an exonuclease, will snip off that nucleotide. Great name, exo, kind of cutting nuclease, cuts the nuclease off, and then we'll flip it back up and we'll start again. So this just shows the detecting the error, the cutting it off, and then it flips back up and starts again. After that, so cut it, boop, flip it back up, and we can add the proper nucleotide. And that is all for this topic. Next, we are going to learn how we make RNA from DNA, and you're going to be introduced to an enzyme that has an equally awesome name as DNA polymerase, RNA polymerase, that works a little bit differently. But here we see that DNA double strand and a single RNA strand being produced from it in the next topics. That's a little preview. So this was DNA replication. Here's our overall process. Here's our subtopics. Good luck, you guys.